And once again, I want to say greetings to each and every one of you. Aquaba, and welcome. Welcome to another round of Culturally Conscious Communications here on the LIV Radio, LIV TV, LivingInBlack.com networks. My name is Kedia Wadu, aka The Conscious Rasta. Weekday mornings, you and I get together live. We come at you from 9 a.m. Pacific time. That's 12 noon Eastern Time, 5 p.m. GMT, and 8 p.m. East Africa Time. We get together with you and we deal with culture, consciousness, historical perspectives, entertainment, economics, education, health, labor, law, politics, religion, family, conflict, future studies, all areas of human interactivity and development. Today's date is Tuesday, the 26th of November, 2024 in the Western calendar. This corresponds to the year 6265 in the Kemetic calendar, to which we have realigned our cultural clock. Welcome to the 7th millennium. You have found your family in a peaceful place on LIV Radio, LIV TV, and LivingInBlack.com. I want to thank those of you who did respond to my outreach, my request of support to help us to stay in the pocket. It's going to be a tough time ahead for all of us. The writings are on the wall. Economically, it's going to be tough. Culturally, it's going to be tough. There is a few slivers of hope with the outgoing presidential regime and the incoming presidential regime, but not for everyone. So we're going to do the best we can to keep focused, to keep together here on LIV Radio. So I want to thank those of you, Brother Lutalo, Brother Myro, Brother Andrew. I am because we are. And also, I had mentioned that this was the worst month of book sales I can ever recall in 30 years. Up until 2 days ago, I had sold one book during the entire month of November, essentially a collapse of my book publishing company, which had been the basis for financing what we do here. So what I'm going to do is I'm going to have a fund drive, a month-long fund drive that begins December 1st. Yes, that's going to happen. We used to do that many years ago, then we stopped doing it. It was exhausting. It was frustrating. But we have to bring it back. We have to try it. I really do not want to give up doing this every day. I don't want to stop. Today's program hopefully will give you a reason to invest in what we call the People's Academy. LIV Radio, LIV TV, LivingInBlack.com, and Black Star Media Global. Today, the subject theme is Stem Cell Glossary. As many of you know, on December 1st, we're going to have a 2-month-long, 56, not exactly 2 months, it's 2 cycles of the moon, 2 moon cycles long, 56-day stem cell rejuvenation program. We're going to be doing this in group, and I'm glad we do have members now joining it. I would like to have 15 to 20 people doing this simultaneously. It is going to be a radical experiment to prove beyond a shadow of a doubt that a natural, holistic, does not breach the skin, natural, holistic fountain of youth does exist. It's the way the body was designed in what's called stem cells. I think the glossary today that I'm going to be sharing will give you a good understanding of exactly what we mean with a real life fountain of youth. So we've got a lot of these different terms we're going to be using today in the glossary, and I have committed this is going to be an upcoming book, and I'm putting that together. I think I may have the book title worked out. You tell me, do you like this? Do I have this? No, I don't have that, but I do have this. Let me just copy, cut, and paste all of this. Copy, cut, and paste that into the book that I'm writing right now. I'm going to style this at normal, paste this, Boom. And bear with me. I did some work in the other room and moved my computer around, and I'm still getting my office back together here. Style this at normal, cut and paste as normal text, and I'll work it out. But I wanted to put all of these into a glossary, and I'm just double checking myself to see how large the glossary is. The glossary, stem cell glossary, style, normal, and put some numbers on that. And the glossary comes to a total of I didn't mean to do all that. Yes, I did. Stem cell glossary, where you is? Excuse me, I'm in the book. Normally I don't mess around like this while I'm in the book. Yeah, the glossary is a total of 50 different terms. Let's go through this stem cell glossary. Again, did I introduce the subject for today? Where am I at? Where am I at? Let me go back to my image and stop the virtual camera. Go back here. My virtual camera shut down for some odd reason. USB camera, there you are. Okay, I'm back. Whew, I disappeared for a second. Okay, bear with me, everybody. We're getting it together. It's challenging right now, no doubt. I'm not the only one that finds myself to be challenged. So let's go into the stem cell glossary. First of all, these are in alphabetical order. Before I go into the glossary in alphabetical order, let me just go right to stem cell and stem cell therapy. Then they'll come up again in the glossary. Stem cell is an unspecialized cell that has the ability to differentiate into multiple specialized cell types. And let's say an unspecialized new cell that will differentiate into multiple specialized cell types, proliferate indefinitely, and maintain its undifferentiated state, making it a vital foundation for tissue growth, development, tissue repair, and regeneration. And stem cell therapy is a treatment approach that uses stem cells to repair or replace damaged tissues or organs, showing promise in various medical conditions, including spinal cord injuries, heart disease, degenerative disorders, ongoing research to enhance its effectiveness. Well, when I put together the list of different applications for the stem cell science, we come to, I think there was a total of about 28 different applications, some of which you will hear as I share the glossary. What is a glossary? A glossary is a spectrum of key words related to a central theme or topic. So that's what a glossary is. And because this area of stem cell science is quite advanced and many of us are unfamiliar with it, we do want to have a working knowledge of the language that we will be using in describing this. These are in alphabetical order. Adult stem cells. These are undifferentiated, also called multipotent stem cells, found in various tissues of the body, including found in the bone marrow and in fat cells, adipose tissue cells, found in various tissues of the body that maintain and repair the tissue in which they reside. They have a limited ability to differentiate into specialized cell types related to their tissue of origin, such as blood cells or skin cells. And just, I would say, also they can differentiate into as many as 200. So they have a limited ability. I'm going to take the word limited out because one cell differentiated into 200 different cells is quite amazing. They have an ability to differentiate into as many as 200 specialized cell types related to their tissues of destination. So I am altering, as you can tell, I am altering the wording here to make it more comprehensive to the audience that I serve. Number 2 on our list of 50, asymmetric division. This is the generation of distinct fates in progeny from a single mitosis, where daughter cells may be positioned in different microenvironments or have segregated intrinsic determinants. Now that definition of asymmetric division, asymmetric means basically they can kind of go all over the place and from a single mitosis. So daughter cells, meaning the destination cells, clone cells from stem cells, may be positioned in different microenvironments. Again, differentiation. Number 3, biobanking. This is the process of collecting, storing, and managing biological samples, including stem cells, for research and therapeutic purposes. Biobanks ensure the quality, accessibility, and ethical use of samples, facilitating advancements in regenerative medicine and personalized therapies. Biobanking. Number 4 on this list, cancer cell of origin. Cancer cell of origin. These are precancerous cell that gives rise to a cancer stem cell, possibly a mutated stem cell or a committed progenitor with acquired self-renewal capacity through mutation. So therefore, we're at that point, the body's moving into cancer proliferation, tumor formation, and metastasis. We don't want cancer cell of origin to exist in our body. The immune system is designed to knock that out. And amongst the spectrum of immune therapies to target and prevent cancer are stem cell therapies. So we'll get into that as well during our study. Cancer stem cell, a self-renewing cell responsible for sustaining a cancer and producing differentiated progeny that form the bulk of the cancer, a critical therapeutic target. So I really could put those two different descriptions, cancer stem cell and cancer cell of origin, put those together, and but for the sake of the glossary, these terms will appear separately, so we'll get a different understanding. Going forward, cell replacement therapy. This is what our bioregenerative program, 56-day bioregenerative program, is going to be about. Cell replacement therapy. Now, but our therapy is natural, holistic, is exogenous, not exogenous, is endogenous, using our own self-manufactured stem cells. So, cell replacement therapy is reconstitution of tissue by functional incorporation of transplanted stem cell progeny. Ours will not be transplanted. Ours will be autogenous, generated by the body. Distinct from bystander, trophic, anti-inflammatory, or immunomodulatory effects of induced cells. Stem cells are used in replacement therapy due to their ability to regenerate damaged tissues and promote healing. Next is cell therapy, treatment method involving the introduction of new, healthy, live cells into a tissue to treat or replace injured or diseased cells, often utilizing stem cells. Going through on our list, and I didn't have these numbered, and I don't want to do all that right now, but we're continuing on our list. Clonal analysis. Clonal analysis is investigation of properties of single cells, essential for formal demonstration of self-renewal and potency in stem cells. Now, I wish I had the mechanism of doing clonal analysis or cellular analysis during our therapy, the only evidence we will have of this will be in tissue reformation that will be obvious. We will see during the 56 days, we will see people's weights shift, moving towards optimized body mass index. People will see their skin clear up and develop. During the time when I first did this on myself, I noticed within the 56 days that the gray hairs in my beard started turning black again. And in the course of 56 days, that is enough time for the hair follicles to get a rejuvenation and for those hair follicles to then produce enough mass and their output growing the hairs that we will see darker hair show up where the hair had been, due to aging factors, had been losing its darker melanin content. So I did see that once before. I expect to see that again. So we talk about clonal analysis, investigation of properties of single cells, essential for formal demonstration of self-renewal and potency in stem cells. We will see this in many different ways, including the reversal of macular degeneration. Some of the people who take this program, who have been reaching for reading glasses, like myself, will, during the course of this, are expected to, it is hoped, will be, I have to be a little bit ambiguous on this language because there are so many variables here, and this is not a real tight, controlled study. This is a demonstration of holistic impact. But some of the people should be able to stop using their reading glasses in the course of 56 days. It happened to me one time before in less than 56 days, in 28 days. So 56 days at a stricter, more comprehensive protocol. Can you imagine? You've been using reading glasses for 5, 10 years, 15 years, and now you no longer have to reach for them. Cloning, the process of creating a genetically identical copy of an organism or cell through techniques such as somatic cell nuclear transfer or natural asexual reproduction. That should be very interesting. I like those topics. The next one on this list, differentiation. And originally putting this program together, differentiation was one of the key words that jumped out at me. I have replaced differentiation with active. No, I haven't replaced it. I think it's still in the title of the book. But going for differentiation, the biological process through which stem cells develop into specialized cells with distinct functions, such as muscle, nerve, or blood cells. Differentiation is influenced by genetic factors, signaling molecules, and the cellular environment, and is essential for forming the various tissues and organs necessary for organismal development and function. Differentiation, one of the three powerful words that I use in building this program. Stem cell activation, differentiation, proliferation, rejuvenation. Those four words serve the core of what this course is all about. Embryonic stem cells, abbreviated as ESCs. These are pluripotent, pluri meaning multiple, potent cells, derived from the inner cell mass of a blastocyst, which is an early stage embryo. Embryonic stem cells can differentiate into nearly all cell types in the body, making them valuable for research and potential therapeutic applications in regenerative medicines. Of course, embryonic stem cells, the use of them in this experimentation drew a lot of castigation back in, I think it was during the Reagan administration. That would be the 1980s, when they were using stem cells, embryonic stem cells, for experimentation obtained from aborted fetuses. And that, of course, drew quite quite a controversy. I'm not going to go into the topic of abortion on today's program. It's a whole nother subject. We're talking about stem cells. But it's understandable how that became so controversial. Epigenetics, study of heritable changes in gene expression that do not involve alterations in the DNA sequence, affecting how cells differentiate and function. And epigenetics is really all about the environment. Epigenetics is really all about the cell specific environment. That's a key point that must be pointed out, that must be emphasized here. Epigenetics is a study of heritable changes, that's our DNA, our chromosomes, in gene expression that do not involve alterations in the DNA sequence, affecting how cells differentiate and function. Epigenetics is really all about the cell-specific environment. Next, I touched on this briefly. Ethics in stem cell research. The moral conditions or considerations, the moral considerations surrounding the use of stem cells, particularly embryonic stem cells. Ethical debates focus on issues such as consent, the potential for life, and the implications of research on human dignity and rights. Ethics in stem cell research. And from what I understand, and Basu Randa, I see you have joined us in the conversation today. Akwawa, welcome. You, beloved, have found your family in a peaceful place. And I always love having you in the house. You have never been afraid to tune into our more science, especially bioscience related broadcasts. I know some people, the language is a little bit intimidating, but nonetheless, there's much that we can do in learning together, growing together, building together. So much that we can do, and which is why we are committed to doing this. So here we go. Continuing, bear with me. Let me save this real quickly. And next on the list, where is it? Here it is. Next on the list, fibroblast. Fibroblast. Fibroblast is a cell that helps form connective tissue in the body, found in many organs, and can be converted into induced pluripotent stem cells. So fibroblast is a cell, part of our connective tissue system. And there's a name for the entire biological system of connective tissues in the body. I can't recall it right now. I'll have to look that up. It's very important, even though none of us think about it at all. But it's underneath the skin, and it connects the entire body. If you can recall that movie Avatar, and they're in the forest and the trees, and they're interfacing with the trees, and all of the trees and the entire forest on the entire planet are all connected with each other. That's what this system is likened to. And I think that probably was modeled after this system of the body. But right now, I hope someone else can recall what is the name of this system of connective tissues. And it's not just connective tissue, but it's a neurological connective tissue, I would say hybrid. But because I can't remember the name, I can't explain it or look up a definition. Maybe somebody else can help me out there. We are up to fibroblast growth factor. We talked about these cells. Now we're talking about FGF, fibroblast growth factor. A family of growth factors involved in a variety of biological processes including the proliferation and differentiation of stem cells. Gene editing techniques such as CRISPR-Cas9 that enable precise modifications to an organism's DNA. In stem cell research, gene editing can be used to study gene function, correct genetic disorders, or enhance the therapeutic potential of stem cells. Now, in the system that we are deploying our 56-day stem cell rejuvenation protocol, we do not engage in any type of in vitro or in vivo gene editing. The only gene editing we will be participating in is through the natural RNA process, where our body responds to environment and then sends signals into the nucleus of each cell saying, well, alter the DNA expression. Don't change the DNA at all. Your DNA is supposed to be pristine from birth. Now, there are people with congenital gene disorders. That is true. But there are also some proposed gene disorders that I don't think are legitimate, such as, I can't remember, is it ALP1? Correct me if I'm wrong. Said to be related to breast cancer. And I can recall in one actress's case, I believe it was Angelina Jolie, who, because her mother and or aunt or two other women close to her had died from breast cancer, she had her breast removed. And I believe, and I'm pulling this off the top of my head, so this may not be perfectly accurate, but I believe that she tested positive for this so-called DNA gene alteration and therefore volunteered to have her breasts removed. Almost went into Dave Chappelle mode in the middle of that, but that's not funny. Not at all. Going forward, genomic modification. Stable, intentional, and direct change of a cell's DNA sequence using biotechnological methods such as CRISPR or gene editing. Again, that's not something that I am interested in, but it is part of the language of understanding the therapeutic and experimental uses of stem cell therapy. Glial cell, a category of central nervous system cells other than neurons. Let me repeat the sentence. Glial cell, a category of central nervous system cells other than neurons, including astrocytes, oligodendrocytes, and microglia. Microglia probably is, making up the majority of the brain. Glial cells essentially translates to gray matter. Let me just put that in here. And that excludes neurons. So neurons, of course, are in these chains, but while there's the glial cells make up the mass. Let me just put that gray matter. Next, haplotype. A group of genes inherited together along the same segment of a chromosome from one parent. Haplotype. Next, hematopoietic stem cells. And this is a very important terminology you're going to hear quite a bit during our studies on stem cells. Hematopoietic stem cells. Hematopoietic stem cells. Cells found in the blood and bone marrow that can form mature blood cells, such as red blood cells, white blood cells, and platelets, essential for the maintenance and regeneration of the blood system. They play a crucial role in the body's immune response and overall blood health. And insofar as the epigenetic environment, that's where it all takes place, in the blood system. So hematopoietic stem cells are very important for our discussion. Next, induced pluripotent stem cells, iPSCs. These are adult stem cells genetically reprogrammed by artificially turning back the clock using the Yamanaka method to an embryonic stem cell-like state, capable of differentiating into various cell types for research and therapeutic uses. Induced pluripotent stem cells offer therapies without ethical concerns associated with the use of embryonic stem cells. Very important information going forward. We are sharing today a glossary of stem cells. I'm sharing it with you as I am sharing it with myself. I'm fairly familiar with many of these terms, but probably close to a third or half of them are new to me and help me to form my overall understanding of what it is that we are about to engage in this 56-day stem cell rejuvenation program. Natural, holistic program. I must emphasize that point. And they had also mentioned in that one the Yamanaka method. That is induced stem cells. And what I have to do is the Yamanaka method is not in my glossary right now. So while I have Copilot open in the background, let me just ask the question, what is the Yamanaka method? Explain in 40 words. So, what is the Yamanaka method? That's going to go on our list as well. The Yamanaka method involves reprogramming adult cells into induced pluripotent stem cells, iPSCs, by introducing 4 specific genes, OCT4, SOX2, KLF4, and CMYC, enabling them to regain pluripotency and differentiate into any cell type. So we're talking about adult cells, not adult stem cells. Adult cells that are reprogrammed using gene editing technologies. Now, while I am not into that, nonetheless, I think it does serve an importance for the purpose of our glossary. So when we come across the Yamanaka method, we'll realize it is a way of taking adult cells, mature cells, and reprogramming them using 4 DNA cuts. Those 4 DNA cuts, very specific cuts, gene insertions, will cause the adult cell to revert to a multipotent or pluripotent cell, meaning it can then differentiate. Now, whether that cell can differentiate many, many times and stick around indefinitely, we don't know. That would be a further question as we would go to look further at that. So we got up to, let me get back in the list where I was. We got up to hematopoietic stem cells, induced pluripotent stem cells using the Yamanaka. That was where we left off. Next, we pick up with macula, a structure in the back of the retina responsible for central vision, containing retinal pigmented epithelial cells lost in age-related macular degeneration, for which stem cell therapy is being proposed as a therapy for macular degeneration. The medical industries want to make a lot of money. For macular degeneration therapy, you're talking about it's going to be on the upper end of these estimates of stem cell therapies, which range from $5,000. I saw one therapy at $2,501. That sounded like something natural to me. can range from 5,000, 10,000, 25,000, 50,000, 100,000 to the most expensive stem cell therapy known, a one-time treatment as $2.1 million. So macula, if we can find a way to reprogram what our body already does and get that therapy for macular regeneration, yeah, that's some good investment. I'm investing there. Mesenchymal. And when I started this glossary, this was the first word that readily came to mind. Mesenchymal refers to a multipotent stem cell that differentiates as progenitor cells for all types of connective tissue, such as fibroblasts, osteoblasts, chondroblasts, and preadipocytes. Preadipocytes. And an adipocyte or adipose adipocyte is fat cell. These cells are characterized by their ability to self-renew and exhibit multi-lineage differentiation, producing an extracellular matrix, ECM, rich in hyaluronic acid. In adults, mesenchymal cells are present in small quantities as part of loose connective tissue, primarily in the umbilical cord, bone marrow, and adipose tissue. So therefore, we can understand why that was the first word I had to look up, because it was coming at me from so many different places. I didn't have a clear understanding of what the word mesenchymal actually stood for. It took me a while to even learn how to pronounce it. So multipotent stem cells that differentiate as progenitor cells for all types of connective tissues. Going forward, multipotent stem cells. You've heard this term used in other descriptions. Multipotent stem cells are stem cells that can differentiate into a limited range of cell types, typically related to the tissue or organ from which they propagate or originate. They play a key role in tissue maintenance and repair throughout an organism's life. So when we're talking about multipotent stem cells, one of the places where we would love to see them and expect to see them show up is in skin integrity, collagen forming in the skin, and the reversal of age-related signs and symbols, such as skin tags, warts, such as moles, such as the darkening underneath the eyes. If my hypothesis is correct, and that's why I'm asking you to join me to test out this hypothesis. If my hypothesis is correct, and we find a mechanism of reversing 1, 2, 3, 5, 10, or more of the 28 different therapies for which stem cell therapies are being proposed, if we find a way of doing this using just natural, holistic, non-invasive procedures, we will have stumbled across the billion-dollar fountain of youth, where people will be able to come, spend 3, 4, 5 days with us, learn these techniques, begin to practice these techniques, take on the habits of these techniques, and then go home and rejuvenate and revitalize themselves. I'm telling you, people, from everything I've studied, that twist didn't work real well. From everything I've studied about longevity, this is probably the pathway by which people will extend human life expectancy beyond its current 78.6 years in the United States to consistently over 100 years, encroaching now upon life expectancies of 135 years. My own particular life expectancy target is 126 years, and I'm very serious about it. But after this next 56-day protocol, I may be able to extend that a little bit even further. Let's see how it turns out. Neural stem cells. Stem cells found in the brain and spinal cord that can differentiate into neurons and glial cells. They are essential for brain development, repair, and potential therapeutic applications in neurodegenerative diseases and injuries, including, and I'm about to pull my book off the shelf as I bring that up, because that goes right into, and I did mention this in my book from last year, Miracle on 2 Feet, Stroke Prevention and Recovery. We're talking about stroke recovery now. And let me send a note to one of my friends. And yeah, you see the book, Miracle on 2 Feet. Send a note to one of my friends whose wife had suffered a stroke. I sent them the copy of the book, Miracle on 2 Feet, and he wrote back and said, man, that book was awesome. His wife, he said, was reading it and was finding it very useful, very informative and inspiring on her pathway to recovery. So now we're talking about people who may have suffered a stroke 10 years ago, 20 years ago, that now have hope because of what I just read, this glossary description, neural stem cells. Stem cells found in the brain and spinal cord. How did they get there in the first place? We found them there. How did they get there? The body manufactured it. Well, if the body manufactured it in the first place, can we revive the process of the body manufacturing stem cells in the nervous system, in the spinal cord? Yes, 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 yes. That's the purpose of this protocol. This stuff gets me excited, y'all. I don't know about you, but I get excited reading about this, learning about this, studying about this. Do I see anybody in our various houses with us today? We are broadcasting live on YouTube. Let me just double check, see if anybody's talking to me there. We are broadcasting live on YouNow, on Rumble. So my YouTube family, nobody talking to me, but I love you. You got nothing but respect for you. Going forward, who's in the house today? Who is in the house today? Talk to me now and hit the like button, if you would, please. So we do see somebody in the YouTube family has hit the like button already, but a whole bunch more people could be responsive. As you can see, Queen Sister Basuranda, that on topics of science, especially science at this level, I get a relatively small audience, and it is what it is. But if everybody out there could at least just hit the like button, let the algorithm know what it is that you're watching today. And I should never have looked at the number of people watching it. It's disappointing because I'm talking about advanced science, glossary. A couple of the books that I put out, I put a glossary in the book because this is so far over people's heads, they won't know what we're talking about. But doggone it, people, this is the real deal. This is the fountain of youth. It's real. It's available. It's designed. It's within our body. Let's continue with our glossary. I'm going off on a tyrant here. We gotten up to neuron. Again, previous we talked about neural stem cells. Now we're talking about neuron. Central nervous system cells that send electrical and chemical signals to each other, forming connections between the brain and the rest of the body, neurons. And again, neural stem cells can differentiate into neurons and glial cells, gray matter. The next term, niche, the specialized microenvironment where stem cells reside, providing essential signals for their maintenance, self-renewal, and differentiation. And when it comes to this specialized microenvironment, we've got to make sure that the heavy metals are detoxed out of that microenvironment. We do know heavy metals have a negative impact on stem cells. Let me just real quickly confirm exactly what I said. I'm going to use Brave here. Brave usually gives me a pretty satisfactory results. We're looking very specifically at the impact of heavy metal toxicity on stem cell rejuvenation. Rejuvenation. So, what is the impact? What is the impact on stem cell rejuvenation? Copy, cut, and paste that. I'm pulling that up on one search engine. This will probably make it into the book. So I'm going to push this up on two different ones. The first one, Brave. Here's what Brave had to say. Heavy metals impact on stem cell rejuvenation. Based on the provided research results, I should have asked for this in 40 words. Heavy metal toxicity can negatively impact stem cell rejuvenation. Here are some key points. This is important. I don't mean to abbreviate this. This is very important. Key points. It lists 7 points. Number 1, heavy metals attach to cells. Heavy metals such as lead, mercury, and arsenic can bind to cellular components, including stem cells, preventing them from performing their normal functions. Therefore, I detox, heavy metal detox, 4 times a year, every year for 22 years now. Point 2, disruption of cellular events. Heavy metals can disrupt various cellular processes, including growth, proliferation, differentiation, damage, repairing processes, and apoptosis, programmed cell death. Point 3, heavy metals related to inflammation and oxidative stress. Chronic exposure to heavy metals can induce inflammation and oxidative stress in stem cells, leading to cellular senescence, meaning the cells go to sleep to die, and reduced regenerative capacity. Impaired autophagy. They inhibit autophagy, a natural process that helps maintain stem cell quality and function, leading to an accumulation of damaged cellular components. That's where we're talking about the end of chromosome strands. What's that called? Begins with TH. But this damages the ability of these cells to replicate themselves clean. Number 5, epigenetic changes. Exposure to heavy metals can induce epigenetic changes, such as DNA methylation and histone modifications, which can alter stem cell gene expression and function. Number 6, reduced stem cell potency. Heavy metal toxicity can reduce the potency of stem cells, limiting their ability to differentiate into specific cell types and regenerative tissues. And number 7, cumulative effects. Simultaneous exposures to multiple heavy metals can have cumulative effects, exacerbating the negative impacts on stem cell rejuvenation. So in summary, heavy metal toxicity can 1, disrupt stem cell function and behavior. 2, induce cellular senescence and reduce regenerative capacity. 3, impair autophagy and epigenetic regulation. And 4, reduce stem cell potency and differentiation potential. These can hinder the success of stem cell-based therapies aimed at rejuvenation and tissue regeneration. Now, that was a critical lesson that we just learned. Now I'm going to do this. I'm going to attach this, go to the end, and attach this to the document that I just created. And did I get it? Did I get it in? Bear with me. Copy. And over in this document, go to the end and paste. Okay, so I did get that in. And it came with some references as well. Good stuff. Good stuff. Going back to our list today, we're going to our glossary. We got up to niche. the specialized microenvironment where stem cells reside. Next, we're talking about oligodendrocyte. Oligodendrocyte. These are glial cells, gray matter, that create myelin, a substance coating neurons and acting as insulation to help them send signals. These are damaged in some neurodegenerative diseases. Yeah, we're talking about the myelin sheath is damaged in diseases such as multiple sclerosis, diseases such as myelin sheath. I know I'm frequently talking about this with clients. The myelin sheath is an autoimmune disorder. That's where I usually will talk about it. Autoimmune disorders, which there are a number of different myelin-related diseases and disorders. See, right now I'm on such a key right now. Just using this glossary lets me know all different areas of research, each of which could be a chapter in the book that I'm writing. So again, we're talking about myelin sheathing, and that is oligodendrocyte, and that's glial cells that create myelin. Now, when we're talking about reversing autoimmune disorders, that's where the target process is our intention. And we can do this using food, because remember what it said about the heavy metal toxicity, about the oxidation and inflammation? Well, these are targeted procedures to correct these problems. We are up to organoids. Organoids are miniature, simplified versions of organs produced from stem cells in vitro, meaning in a test tube, laboratory. These are used for research, drug testing, and modeling diseases. Organoids. So basically they are synthesized organs, miniatures. Next, pancreatic beta cells. This is very, very important, especially when so many people today are diagnosed with diabetes. Pancreatic glial beta cells helping the pancreas produce insulin. These are attacked by the immune system in type 1 diabetes, and type 2 adult onset diabetes mellitus is also an involvement of the pancreatic beta cells, and the insulin that's being produced is actually not being received throughout the body, ushering fat into different cells, storage cells. So, very important. Parthenogenesis. Activation of an unfertilized egg by chemical means, causing it to begin dividing as it would after normal fertilization, potentially used to derive embryonic stem cells. That's parthenogenesis. But remember, this is chemical stimulus, whereas in nature, this division in the cell is caused by the chromosome insertion, including the XX or XY chromosome the man puts in to determine whether this will be a female or a male. So without the 23 pairs of a 46 whole chromosome presence for a developing embryo, without the 23 pairs contributed by the sperm cell, then that clone cannot develop adequately. Now, I'm not going to go too deep into that topic for today. I think it might be a little distracting. Human cloning has been banned internationally. Animal cloning is still taking place, but it's, from my understanding, it's not very successful for most species. And let's go forward. We gotten up to, that was parthenogenesis. The next one, plasticity. Ability of stem cells to adapt and differentiate into different cell types under specific conditions, highlighting their potential for various therapeutic applications. And again, when we're talking about plasticity earlier, we talked about how stem cells proliferate in the environment in which they are created and differentiated. This environment is taking us back to the word epigenetic. We talked about that earlier. The epigenetic environment, pluripotent stem cells. This is a very important topic. We've mentioned this several times. Pluripotent stem cell, early stage cells that can become any type of cell in the body, including embryonic stem cells and induced pluripotent stem cells. So induced pluripotent is edited, gene edited. That's CRISPR. But embryonic stem cells are natural at the beginning of the life process. When sperm meets the egg and they conjoin, right there is when the pluripotent stem cell process begins. The embryo starts off just as a cluster of, I believe, 700 or so, and I may be crossing numbers up from different things. A cluster of 700 stem cells is where embryonic life starts to differentiate. And then later on in adult stem cells can differentiate into 200 different types. Progenitor cells. These are intermediate cells derived from stem cells that are partially differentiated and have a limited capacity for further differentiation. Progenitor cells. So in this case, progenitor cells are stem cells that are now specifically targeting liver, say liver stem cell growth, liver cell growth. They may be targeted at cardiac, specific organ types. These are progenitor cells already differentiating. And they probably can be obtained from bone marrow or adipose tissue in that matter, or obtained from spinal cord. Regenerative medicine, a field of medicine focused on repairing or replacing damaged cells, tissues, or organs using techniques such as stem cell therapy, tissue engineering, and gene editing to restore normal function and improve patient outcomes. But let's be very, very clear. Regenerative medicine is functional medicine. Regenerative medicine is clinical nutrition medicine. Regenerative medicine is holistic medicine. It is natural medicine. All of these are processes by which we repair or replace damaged tissues, organs, using natural techniques, not just using allopathic medicine. That's very clear. Do not allow, we will not allow, I will not allow the mainstream allopathic medical community to appear or attempt dominating control over the processes of stem cell therapies. Because the reality is, since the dawn of life, primate life and above, stem cells have been there all along. So Western medicine is relatively recent even in discovering this. This is a cutting edge therapy that they're talking about right now. And I can appreciate the scientists on the front edge of allopathic medicine for exploring this. I can certainly appreciate it. That's why I read their papers. I read their documents. But my intention, my bias is always going to be that nature can do this using what she provided us in our environment, in our food, in our exercise, our rest, the 7 principles of optimal health. Every breath I take feeds the process by which I generate vital new cells. 300 million per minute. That's a pretty awesome process. So I'm going to stick with Mama Nature on this one. Self-renewal. Again, from the definition of regenerative medicine, the next one on the list is self-renewal. The ability of stem cells to undergo numerous cycles of cell division while maintaining their undifferentiated state, crucial for their role in tissue maintenance and repair. Self-renewal. Next on our alphabetical list, our glossary, somatic cell nuclear transfer, SCNT. Somatic cell nuclear transfer is a cloning technique where the nucleus of a somatic cell is transferred into an enucleated egg cell, creating an embryo that can develop into a source of stem cells, potentially useful for therapeutic cloning and research. So when we're talking about a somatic cell, that's an existing cell, an adult cell, where the nucleus of another cell whose differentiation is preferred is inserted into the somatic cell to replace its nucleus, and therefore it becomes a clone. Stem cell. Again, we started off the discussion with this description. It's in our glossary under S. Stem cell. A stem cell is an unspecialized new cell that has the ability to differentiate into multiple specialized cell types, proliferate indefinitely, and maintain its undifferentiated state, making it a vital foundation for tissue growth, development, repair, and regeneration. And put a comma after tissue growth. Stem cell culture, a laboratory technique used to grow and maintain stem cells under controlled conditions, providing a platform for studying their properties, behavior, and potential applications in research and therapy. Next, stem cell niche. Again, we talked about niche, the specialized microenvironment surrounding stem cells that provides essential signals for regulating their self-renewal and differentiation. The niche plays a crucial role in maintaining stem cell populations and ensuring proper tissue function. That is almost the same description as the one we used for niche before, and I think I'm going to remove that other one since it was much shorter and didn't really contribute to the same understanding that stem cell niche contributes. So that other definition we read earlier was included in this one, but this one is much more specific to our glossary. Stem cell therapy, a treatment approach that uses stem cells to repair or replace damaged tissues or organs, showing promise in various medical conditions, including spinal cord injuries, heart disease, and degenerative disorders, with ongoing research to enhance its effectiveness. And again, in the book, I identified some 28 different targets for stem cell therapy. And let me just add that there are dozens of targets for current stem cell therapy and research. Teratoma, a type of tumor that can contain various tissue types, often formed from pluripotent stem cells where they differentiate uncontrollably. Teratomas are used in research to assess the pluripotency and differentiation potential of stem cells. Now, teratoma, terra relates to terrain, meaning environment, in geography. Toma relating to tumors, which often are related to carcinomas, cancer tumors. So when we're talking about teratoma and this uncontrollable differentiation, to me that can be useful when we attempt to explain the cause of cancer. There is no single cause of cancer. There are multiple causes of cancer, including one of my favorite known causes of cancer is the mutation of eukaryotic cells to prokaryotic, from aerobic to anaerobic, from natural ATP production in the mitochondria to unnatural energy production using fermentation in the cell. But those are just one. That was 1, 2, 3. But there are many things that trigger these, including exposure to chemicals, including degeneration of the immune system. So there are multiple causes. So this idea of teratoma, a type of tumor that contains various tissue types, often formed from pluripotent stem cells that differentiate uncontrollably. Our purpose of our investigation is how do we best naturally, holistically control stem cell activation, differentiation, proliferation, and the other Asian, which was earlier, and I can't remember it right now. I've been using those three so much in writing that now I have to add that fourth one. We're in the T's now. In the alphabetical order of our glossary, we're up to tissue engineering, an interdisciplinary field that involves biology, materials science, and engineering to create functional tissues or organs for medical applications, often utilizing stem cells to enhance tissue regeneration and repair damaged or diseased tissues. Now, I know tissue engineering is very important right now for applications in traumatic injuries, such as what a soldier might go through injured on the battlefield, but also for burns. It's a critical area where this research has been being driven by repairs to burned tissue for wound healing. Very important. And I can only imagine, as this research and these therapies are being developed, I can only imagine that perhaps in 10 years, 15, 20 years or so, if a person has a major traumatic injury to the skin, that they will be put into a therapeutic scenario, whereas their body will rapidly, assisted by bioengineering technologies, will rapidly replace the damaged skins. This research is also looking into things where when people have had traumatic injuries to their face, such as I know in one case where I read about this particular therapy being developed was a woman who had her face bitten off by a chimpanzee. This was in more than one case. I can recall studying this. People, they get all friendly with chimpanzees, not realizing chimpanzee is kind of PO'd all the time at you for taking it out of its natural environment and raising it, thinking you're raising it as a child. The chimpanzee reminds you just how messed up children can be, but the only thing is a chimpanzee might be 8, 10 times stronger than a human when their bite is quite damaging. And people had their face ripped off by chimpanzees using these type of, what was the word we're looking at? These type of therapies, tissue engineering therapy, to repair damaged or diseased tissues. Very important. We're up to therapeutic cloning, a process aimed at creating embryonic stem cells for medical treatment by cloning an organism's cells, leading to the development of patient-specific stem cells that can be used for regenerative therapies without the risk of immune rejection. Now, when we were talking about the cost of stem cell therapies that are out there, and that the most expensive one was $2.1 million for that therapy, this is what they're talking about. But in this case, they're not using autogenous cells. They're using cells from someone else being donated. And there are different stages to this as well, because before implanting the new revitalized stem cells, they will quite often radiate the bone marrow and take the bone marrow out, take it out, and then replace it with this new therapy. This is radical stuff, y'all. If we can avoid all of that, it would be in our best self-interest. We do know that to be true. totipotent totipotent stem cells, totipotent stem cells, the most versatile stem cells that can develop into any cell type, including both embryonic and extra-embryonic tissues like the placenta. They are present only during the earliest stages of embryonic development and can form a complete organism. So we're talking about totipotent stem cells. These are the stem cells that proliferate when the sperm cell and the egg conjoin. That is when totipotent stem cells are activated, and they can complete the creation of a whole human body with the ability to regenerate itself and replicate itself in future generations. That's toti, meaning all of potent potential. Transdifferentiation. We talked a lot about differentiation. Well, what is transdifferentiation? The process by which one specialized cell type is converted directly into another without reverting to a pluripotent state. Transdifferentiation offers potential for regenerative medicine by generating specific cell types for therapeutic applications. We got two more of these 50 definitions in our glossary. The next is umbilical cord blood stem cells. Umbilical cord blood stem cells. These are hematopoietic stem cells collected from umbilical cord blood at birth. Again, these are related to the totipotent stem cells 2 definitions ago. The umbilical cord is, of course, where the blood between the mother and the birthing baby is at the highest integrity, not exposed to the toxicities of the world. These are hematopoietic stem cells, meaning derived from blood and bone marrow. And these hematopoietic blood stem cells are collected from umbilical cord blood at birth. And for most people, when a baby's born, what do they do with the umbilical cord? They give Papa a pair of scissors and say, "Would you like to cut the umbilical cord?" And Papa says, "Yes." And they take that umbilical cord out of the room. One can only imagine what happens to the umbilical cord after it's removed. Let me just ask this question. Maybe we'll get an answer. I like that last answer we got there. What happens to the umbilical cord after birth? Let's ask. I'm asking Brave, what happens to the umbilical cord after birth? Point 1, cutting the cord immediately after birth. The umbilical cord is clamped and cut by a healthcare provider, leaving a short stump attached to the baby's abdomen. 2, natural drying and separation. The stump will naturally dry up and fall off within 1 to 3 weeks after birth. Number 3, care and maintenance. During this time, it's essential to keep the stump clean. Number 4, natural separation as the stump dries. But important, let me just ask this question. Is umbilical cord Is the umbilical cord after birth ever used in medical research and therapeutic development? See, with AI, you got to be careful how you ask the question. I asked a simple question, what happens to the umbilical cord after birth? And they just cut the cord and just went to the stump. But now I'm asking this question. Here is the question that I asked. What happens to, let me read this real careful. What happens to the umbilical cord? Where is it at? Yeah. Well, here it is. I thought I had the question here. What happens to the umbilical cord after birth? And the end of that question, I can't find the question, but you heard me ask the question. I'll be able to repeat it. Here's what it says. The umbilical cord is a rich source of stem cells, which have shown great promise in medical research and therapeutic development. Research is focused on the use of umbilical cord-derived mesenchymal stem cells for the treatment of various diseases and disorders. Therapeutic potential of umbilical cord blood-derived mesenchymal stem cells have been shown to have a broad and efficient therapeutic capacity to treat various diseases and disorders, including autoimmune disorders, cerebral palsy, type 1 diabetes, myocardial infarction, neonatal morbidities such as bronchopulmonary dysplasia. So they listed 6. My book lists 28. Not to take away from them, but to say that we got to go below the surface when we're engaging with AI. Couple more points. Clinical trials and research. Several clinical trials and research studies have been conducted to investigate the safety and efficacy of umbilical cord-derived mesenchymal stem cells for the treatment of various diseases and disorders. That's just repeating itself. Future directions and challenges. While the therapeutic potential of umbilical cord-derived MSCs is promising, there are still several challenges and limitations that need to be addressed, including 1. Standardization of MSC isolation and expansion protocols. 2. Development of effective delivery methods. 3. Ensuring long-term safety and efficacy. But they didn't mention anything about any ethical challenges. Remember, that was in our glossary up in the E's. I'm going to return to that in a second. Overall, the umbilical cord is a valuable source of stem cells for medical research and therapeutic development, and ongoing research is focused on unlocking its full potential. So what happens when your baby's born in a hospital? They give you the scissors, cut the umbilical cord. Now you're focused on the baby, the stump, the mama, and everything you should be focused on. But somewhere in the back of the room, somebody walked out of that room with that umbilical cord and maybe the placenta and said, "We about to get paid." That's just hypothesis on my point. But we do see they are using these materials in their therapy. We've gotten up to the last of our definitions, and we talked about this one a little bit earlier, but I inserted it. Yamanaka method involves reprogramming adult cells into induced pluripotent stem cells, iPSCs, by introducing 4 specific genes. This is gene editing. OCT4, SOX2, KLF4, and CMYC, enabling them to regain pluripotency and differentiate into any cell type. Now, from my understanding, in the allopathic mainstream medical field, the Yamanaka method is awesome. It's just awesome. And in reading the name of this Japanese doctor who developed this technique called the Yamanaka method, everything I read in the mainstream research about mentioning him were glorifying this technique. So I'm not going to be what's the word? I'm not going to be negative about him. It's a specific something downer Debbie or something. I'm not going to be the downer Debbie here. No, I think that the scientist should be acknowledged for his accomplishments, his breakthrough. That's quite radically different than what I do. But what I do is non-invasive. I do not take test tubes and put tissue materials out of your body into the test tube and then put that back into your body. What I do is discover all of the ways that nature gave us this beautiful, beautiful bounty. by which we could enjoy healthy longevity. But unfortunately, mankind in his limited experimental behaviors has altered the patterns of nature too many times and created toxic environments that do not work well with the natural order, the natural plan. Hey, listen, I hope you agree with me that today was a good program. I do see my beloved Brother Kwame Reggie is in the house. I see you. He says, greetings, Robert Keedy. I just joined today's broadcast, and I am looking forward to your upcoming stem cell class starting on December 1. Yes, Kwame Reggie is in this class. Kwame Reggie, do the best you can. If you can convince other people to at least take a look at what it is that you are committed to. Now, it might be easier after they see the results of your 56-day transformation. I think I'm going to be teaching this class again and again, and I can promise everybody out there I will not be teaching this class at this low, low, low price. Now, I have done this on myself. I know it works. I know it's non-dangerous. I did not get sick, did not have any negative responses, reactions during the 56 days in March 2021 when I completed this 56-day protocol. I had the before and after picture. I think, Brother Reggie, you've seen that. I cannot and will not guarantee that other people will not have any negative side effects. I can't guarantee that. I can't guarantee that anyone will follow the protocol just exactly as I teach it, instruct it, and I myself doing it along with you this time. Things could happen. I mean, a piano could fall off a second story of a house and scrape your head and you get an infection. They say, well, had he not been doing this 3-day water fast, he would have been walking out the house at that same time of day. I don't know. I'm just kind of reaching over here. But what I can say is this. This is absolutely brilliant, brilliant science. I'm very proud of it. Now, there was one other question. The last thing we did, and we read this into the record, heavy metal impact on stem cell regeneration. And I think I'm going to put heavy metal and stem cell into the glossary. Somehow it'll be in there. I think it was already in there when we talked about environment and maintaining the optimized environment for the stem cells, a microenvironment. There was a particular term. Hey, I'm going to have to go through this glossary a whole bunch of times myself to be able to utilize this language freely. That's what a glossary is for. You read it again and again and again. When you're going through the book, you hear these words. I recognize that word from the glossary. Let me flip to the glossary real quickly and get that word into the definition. I probably will be adding a number of other terms into this glossary as well. That's what a glossary is useful for our understanding. Wow. I didn't know if I would be able to fill up the entire program. We're up to what, 85 minutes now? 83 minutes? I didn't know if I would be able, no, not 83 because I started about a little bit later. But I didn't know. But now we do know. And so therefore, I just want to thank you all for tuning in to LIB Radio, LIB TV, and livinginblack.com. I want to thank you all for being a part of something that means a lot to me. I want to thank you all for your support too. It's not too late to join the class. The class begins on Sunday, the 1st of December. 56 days. So it takes us through about the third week or so in January. During this detox fast, you will be experiencing your holiday season. If this goes the way I anticipate it'll go, you will notice, like, unlike everyone else around you, this holiday season, you will not gain weight. Your weight will normalize, will balance. If you're underweight, you will increase your weight. If you're overweight, you will decrease your weight. That's based upon the optimized body mass index, which I encourage everyone out there, please be aware of BMI and go for it. We do know that optimizing your BMI adds a minimum, I believe it's 7 years to your predicted life expectancy. Who couldn't use an extra 7 years just by not being too heavy or too light? You've been tuned into your favorite. We aim to be your favorite. We are LIB Radio. We are LIB TV. We are livinginblack.com. Today we've been talking about stem cell rejuvenation, integrity, revitalization, differentiation. And we've been talking about it, and proliferation, talk about in the context of this new language that we're going to have to master to be able to get this into our life. You've been tuned into LIB Radio, LIB TV, and livinginblack.com. Tell your friends to tell their friends that we have our own now. And we're very, very proud of who we are, what we do, and where we are going with our destiny. Peace and prosperity to each and every one of you on LIB Radio, LIB TV, on livinginblack.com. Bumper music, take me out now. And let me show you the books in case anybody wants to support what we do. It is the book sales that supports us to a great extent, has been, but we've been very disappointed. The month of November, the worst month of book sales I can ever recall in 30 years. Can we do better than that? We had better do better. Because if we don't do better, we're going to lose something that may be very important to each and every one of us. You can get the books at keidi.biz, Keidi.biz. Peace and prosperity to each and every one of you.